Top Bed Talk.
Monty Mython here. As we prepare to move into the next phase of the COVID crisis, we're going to be widening our focus here on Top Bed Talk. We will continue to deliver COVID-specific programs, but we all have a huge additional responsibility as we try to reboot normal services. So although this piece is not directly related to COVID-19, we believe it's information that is crucial to the bigger task of rebuilding the healthcare system as we learn to adapt and live with this new virus. Thank you to our sponsors and to you, our listeners, for helping us to share this important information.
Hello, I'm Geoff Lacey and welcome to Top Med Talk, and this is our special series of podcasts on Xtreme Everest, the groundbreaking research program that for the past decade has pursued a novel approach to scientific exploration, performing large-scale healthy volunteer studies at high altitude to mimic the effects of critical illness, with the hope that their findings may be brought from the mountainside to the bedside and improve care for our sickest patients. In these podcasts, we'll take you through the story of Xtreme Everest, from its inception to its discoveries and to the future of this unique endeavour. I am very pleased to be welcomed by Dr. Andrew Murray today, reader in metabolic physiology at the University of Cambridge. Andy, many thanks for joining us.
Thank you.
Now, you've been involved in both of the Xtreme Everest expeditions, which have had the broader objective of trying to work out how the human body responds to hypoxia, and particularly how those that respond well respond to hypoxia. But your work has focused on mitochondrial function under these conditions. I think maybe it's helpful, first of all, if you could just explain to us what a mitochondrium is.
It's a good place to start off. Well, the mitochondria exist in most cells in our body, not red blood cells, but that's a real exception. So the mitochondria are thought to originate from independent life forms, so by bacteria that underwent a process called endosymbiosis, whereby they were engulfed by a host cell. And what perhaps looked a little bit like a parasitic relationship in the first place has become a beautiful symbiotic relationship. And mitochondria, which over the years have permitted the evolution of multicellular life, provide the vast majority of the energy that we use to drive the processes of life. So essentially these are unique biochemical environments within the cell, metabolic pathways that are unique to the mitochondria, consume most of the oxygen that is consumed by our body, and in doing so produce a molecule ATP from the phosphorylation of another molecule ADP. That process requires energy, so the energy comes from the oxidation of fuels such as fat, glucose, ketone bodies, even alcohol. And the ATP is the currency that we use to drive the process of life, whether that's movement, growth, reproduction, energy for our brain, our heart, and all of our vital organs.
So they are the factories, if you want, of the body. So using the oxygen as fuel to produce the energy that keeps us alive.
Yes, yeah, and often termed the powerhouses of the cell, so the furnaces in where the fuels are burnt with oxygen.
Tell me about the initiative you had to get going or get involved with the Xtreme Everest team, and particularly to focus on the mitochondria. What was your kind of inspiration?
My early inspiration really came from Hugh Montgomery, who was leading the science program for the first expedition. Hugh was my PhD examiner, and the first expedition really came along at a great time for me. I'd been working on mitochondria in the context of heart failure, so a condition where we see that the mitochondria are dysfunctional. We know that there are many stresses going on in the failing heart, and one stress that's been implicated is hypoxia. So understanding how hypoxia modulate mitochondrial function was, I suppose, already an interest to me. I just hadn't thought about applying it at altitude, and it was Hugh who invited me to get involved and get thinking about what I could contribute to the first expedition.
And so that leads nicely into what you did on the first expedition. What was your objectives and your findings?
So I really joined an expedition that was already very well planned. There was a clear plan about the route that the team were going to take to base camp and to the summit, what the subject profile might look like. So I had to work within some constraints, but we decided to employ techniques with a minimum sort of impact on the existing expedition plan. So we were using a technique called NMR, nuclear magnetic resonance, to look at heart and look at muscle. The issue with NMR is that you rely on enormous pieces of equipment weighing several tons, scanners, very large magnets with a supply of liquid nitrogen, liquid helium. This is not something you could take to base camp. So the idea was that we'd study people pre and post expedition. In addition to that, and talking with Danny Levitt, who was one of the science leads for the first expedition, we decided after much toing and froing that it was definitely worth us taking skeletal muscle biopsies from some members of the team if we could. And that way we can look in a bit more detail at the mitochondria and some of the molecular pathways. But we would take those biopsies in the field, freeze them, fly them back to my lab to study.
And were you doing those biopsies on all the volunteer trekkers, or was this a particular group that you focused on?
It was a small cohort. We didn't fancy trying to get that through the ethics. And, a biopsy is a reasonably invasive procedure as well, so I'm not sure everybody would have agreed to it. We looked at a small group. We looked at some of the lab staff who were planning to be at base camp for the duration of the expedition, but also the summit team who gave us biopsies before the expedition in London and then after they returned to base camp. So the biopsies were taken at base camp and frozen and flown back for our analysis.
And what did you find then? What was the learning points from that first expedition?
Well, the initial results really came from the NMR studies. Because we could acquire that data, we could analyse it fairly quickly. What was interesting was looking at the hearts of these subjects, and we use an index which tells us something about the energy reserve in the hearts. We're looking at a molecule phosphocreatine, which is the stored reserve of energy. And we found this energy reserve was falling in all the individuals. And this wasn't just the people who were heading to the summit of Everest. And you might expect it there. You're really stressing their bodies with quite profoundly low oxygen levels. But also the people who are coming back from base camp. So energy levels are falling. What was striking about that was that, okay, you might expect them to have lower energy levels at altitude where there is less oxygen available to them. But they were returned to sea level. They were breathing sea level air. They spent time at altitude. They had more red blood cells. You might think this is a positive experience to them in terms of the way the body is acclimatizing, and possibly they would have higher energy reserves upon return to sea level. This is the principle behind altitude training, of course. Whereas actually what we found was that these energy reserves are falling in the hearts of all the subjects.
Over what time period did you then see any kind of resolution, or was there resolution?
Yeah, we followed them up 6 months later. We probably should have had a few measurements in the meantime, but this was a striking finding. We got an amendment to our ethics to get them back in. As soon as we could get everybody back was 6 months later, and thankfully we'd all recovered. So thankfully I was one of the subjects. So our energy reserves had returned to normal in the hearts.
And so what was your conclusions to that? What was your impression as to what was going on physiologically?
Yeah, I felt there was metabolic reprogramming. So those metabolic pathways in the heart were being adjusted. And we know the heart is metabolically flexible. It can use fatty acids as fuel. It can use glucose as fuel. It adapts to the fuel use depending on circumstance. So we see in the failing heart, for instance, there's a reprogramming away from the use of fat towards glucose, which is more oxygen efficient. So our feeling was that the metabolic machinery of the heart had been altered. Now, of course, we didn't have heart biopsies for these subjects. So
that would have been difficult for ethics.
That would be even more invasive than the skeletal muscle, and it was hard enough to get them to agree to that. But then we were able to go back to the skeletal muscle biopsies, which we had preserved at this point, and we hadn't touched them.
And what did you find with the skeletal muscle biopsies?
Well, we didn't see a lot in the team at base camp, so there didn't seem to be a lot happening in the early stages of time at altitude. But in the group that had summited Everest, they had a more intense hypoxic experience, but also a longer experience by this point. We found that they'd lost about 20% of their mitochondrial density in the muscle.
So they'd lost 20% of that component that is dependent for making energy.
Yes, exactly right. So it seemed that their muscle was decreasing its capacity to use oxygen. So they're spending time in an environment where there's less oxygen available. And looking back through some of the older textbooks, the suggestion was that you'd perhaps make more mitochondria.
Yeah.
But then what's the point in having more if you're supplying them with less oxygen? So it seemed that instead the muscle was matching up oxygen supply and demand in quite a clever way, really.
And so with those findings, how do you then develop the plan for the Xtreme Everest 2 expedition in 2013? You went back with them, but with a new plan of attack, so to speak.
Yes, it was, yeah. And we also got a few hints from that data that it wasn't simply that there were fewer mitochondria, but there was a specific downregulation of the capacity of the mitochondria to use fatty acids in particular, perhaps in favour of glucose and other substrates. So the idea was that we dissect that mitochondrial function in a bit more of a delicate way. And this time we were very much involved in the planning of the trip. And the idea was that we take with us some equipment that could actually, rather than just snap freezing the muscle biopsies at base camp, we could actually put them into our machines, measure their capacity to use oxygen under different circumstances, and really probe how finely tuned they were to.
So on site, you were getting that kind of information?
Yeah, absolutely right. It's difficult now to really understate how challenging this is. We run this technique in my lab all the time, but I've got a very well-stocked, controlled laboratory in Cambridge. If I need reagents, I can call up Sigma and they'll send it the next day. This is much harder in Basecamp to get any equipment working, really. To get it up there, it's got to be carried in by yaks or by Sherpas. And so to make these measurements was a real challenge.
And were you able to get all the measurements you wanted?
We did. And it wasn't without challenges. We had to source reagents at the last minute in Kathmandu, so there was some issues with one...
Last-minute stress?
Yeah, one of the barrels had a bit of a leak on the way to Basecamp, so we had to source alternative reagents. But we were able to make all these measurements, and in a large part that's a tribute to the two PhD students, James Horscroft and Alex Kopvicker, who were running these experiments at Basecamp.
And this time, as in for the second expedition, you weren't just looking at the trekkers, you were also focusing your efforts on the Sherpa people, those that we've said several times that they seem to be the best adapted people on the planet in terms of coping with hypoxic conditions. So you were able to also get muscle biopsies from them, I understand.
Yeah, absolutely right. And I mean, the Sherpas were big motivation for getting involved in the second trip. When we came back from the first expedition, we would give many talks to members of the public, to the scientific community. And in almost every talk, we were asked about the highland populations and how they were adapted to altitude. So the ability to study the Sherpas was a big plus for us. And there's a few clues that there was something going on in their mitochondria. I mean, for one thing, and it sounds paradoxical, the Sherpas actually have a lower concentration of red blood cells at any given altitude than lowlanders. So we go to altitude, we make more red blood cells to carry more oxygen to the tissues. In the Sherpas, it increases too, but not to the same extent. They are slightly suppressing that response, which doesn't sound quite right at first. There are benefits to having slightly fewer red blood cells. Your blood is less thick and it's easier to transport it around the body. And we see in the Sherpas that, for example, blood flow through the forearm and through other limbs is better than lowlanders. So they've got fewer red blood cells. There are clear benefits, but this does mean they've got less oxygen in their blood than we do.
And yet they perform at such a higher level.
Yeah.
Your hunch was that was through a modification of their mitochondria.
Yeah, it was yet again a clue that it's not just about oxygen delivery, it's about oxygen utilization. The other clue for us were a number of studies that were coming out around that time, pinpointing variations in the genetics, in Sherpas, in other populations in Tibet. A number of these gene variants were coming up time and again, but one that was really interesting to us is a gene called PPARA, which won't mean a lot to the listeners necessarily, but it encodes a protein that's the master regulator of fat oxidation in heart, in muscle, in liver. So given our previous hints that fat oxidation is modified at altitude, it was very interesting to us that the Sherpas had a variation in the gene that is a major regulator of the muscle's ability to use fat.
And explain why modifying your ability to metabolize fat may or may not be an advantage.
Right, so fat's a great substrate. Most of our body's fuel reserves are in the form of fat in our adipose tissue. It's the main fuel used by the heart day to day. It's used by muscle. It's used by other tissues. So fat is not a bad substrate. In terms of the ATP and the work that you can produce per gram of substrate, it beats glucose hands down. The problem with fat is that it is relatively oxygen hungry. So in order to release that ATP, you need to use more oxygen than you do if you're burning glucose. So under situations where oxygen becomes limiting, so hypoxia is one example, exercise might be another, certain clinical conditions, we see tissues switching towards glucose use. Now, they may not get the full yield of ATP, but they're doing it in a way which is using oxygen more sparingly. So given that the Sherpas possibly had and ultimately did have a different capacity to use fat, the suggestion to us was that they were dialing down the fat metabolism in favour of more energy-efficient substrate.
And so you alluded to some of your results there. Can you expand on that? What did you find on the second expedition with the muscle biopsies and the other metabolic studies you were doing?
Yeah, so this gene variant, it thankfully was enriched in our Sherpa population. As I said, it hasn't been seen in all groups in Tibet, but the Sherpas do happen to have this variant. It's associated with lower expression of this gene, and what that means is they've got a lower capacity to use fatty acids. When we probe the mitochondria a little bit further, we can measure the mitochondria using oxygen to make ATP, but also the way they use oxygen for purposes other than to make ATP, an inefficient use of oxygen, if you like. And this was much lower in the Sherpas than the lowlanders, even at baseline. So the genetics, these were not Sherpas who were living at altitude. They hadn't been at altitude for 9 months prior to the expedition. And yet within their genetics, they retained this lower capacity for fats and a more efficient metabolism.
And were those changes evident when you looked at them at sea level, at lowland, or was it only something that became evident at altitude?
Yeah, we studied them in Kathmandu rather than flying them to London. I mean, it's their home altitude. But the differences were already there. They were more efficient than our lowlanders and had lower fat oxidation capacities. When we took them to altitude, there was very little change, actually. They didn't show any responses in terms of the fat oxidation. It was absolutely the same in terms of the mitochondrial capacity, the leakiness of the mitochondria. It was all very similar.
And then what about the biopsies that you took? What did you find with that?
Yeah, these were the measurements of the mitochondrial function in the biopsies. We flew frozen sample home and then looked at levels of metabolites, levels of these metabolic regulators, and it all added up to this picture of lower fat oxidation. In the lowlanders, interestingly, we studied them both at low altitude and high altitude, and their pattern of metabolism seemed to converge to some degree on the Sherpas, particularly as they spent more time at altitude. We studied the group after 2 months at base camp, and here we are beginning to see signs that fat oxidation is being down-regulated, that the mitochondria are becoming efficient. So,
but anywhere near the kind of Sherpa levels?
Not quite to the same degree, certainly with the fat oxidation. And it was associated with some negative consequences in the lowlanders. They weren't burning the fat, they weren't using it. And so the muscle was accumulating some of these fatty acid metabolites they were building up in the muscle. Curiously, in the Sherpas, we didn't see that. It suggests they got some other way of disposing of the fat.
And on the first expedition, you mentioned how the summiting team showed in their muscle biopsies the number of mitochondria had decreased. What were your findings with this second expedition in terms of the numbers of mitochondria?
Yeah, we didn't use the sort of imaging technologies that we'd used on the first expedition, so we couldn't say absolutely that they'd lost or hadn't lost mitochondrial volume density. The impression that we get from the data, so we looked at capacity to use oxygen, for instance, we're looking at surrogate markers of density. They didn't look desperately different, actually, but of course this wasn't a group that had summited Everest. They hadn't been to that extreme altitude. So I think the take-home message from this is that it's not all about how many mitochondria you've got, and although if you go to extreme altitudes you might see that you're losing mitochondria, you can get more subtle regulation of the metabolic pathways within the existing population.
And how do you see, or do you think it's possible that we would somehow be able to manipulate non-Sherpa people to develop, or at least adapt their mitochondria to become more efficient?
Well, I'm an optimist, so I would hope so. You know, it's obviously a goal of the whole Xtreme Everest expedition to translate these findings into better clinical outcomes. I've got a project running at the moment with Dan Martin, who's been one of the scientific leads all the way through and led the 2013 expedition. Dan's day-to-day job is on an intensive care unit at the Royal Free Hospital in London. Along with Dan's fellow, Helen McKenna, we're looking at muscle biopsies from patients who are admitted to ITU. And what we see are very similar patterns of change. The same targets come up in these patients as we see particularly in the lowlanders at altitude. So fat metabolism seems to be dialed down in these patients. Certain sort of mitochondrial targets seem to be also affected by admission to ICU. And then we see a different pattern of change as they move from early critical illness, so 2 days in the ICU, onto 7 days later. And I suppose the goal would be to understand, the goal is always to understand who's doing well, who's doing less well. At altitude, that would seem fairly clear who's doing well, who's doing less well. It's even more clear when you get to the ICU. Clearly those that survive are the ones who are doing well. So trying to understand if there's a distinct metabolic profile to the patients who survive in compared with those who don't survive would be the next step. And there's early indications that, particularly as you move from that 2 day to 7 day time point, that these patient groups are moving in opposite directions.
I suppose those findings are a great illustration about how these expeditions to high altitude, it's a natural laboratory in which we can start to unpick all these quandaries about hypoxia and how the body adapts to it.
Yes, absolutely right. And I think these expeditions have been a wonderful way to bring colleagues together who wouldn't necessarily work together. I'm not sure I would have encountered Dan had it not been through these expeditions, but of course, and I'm not sure he would have been aware of the work that we do. But by bringing researchers together in this integrated way, this word keeps coming up, integrative physiology, and understanding that, the whole is greater than the sum of the parts, all the mitochondria, you can't look at them in isolation. It's also about the microcirculation, it's also about oxygen delivery, and trying to understand how all these pieces fit together is very important.
A true collaborative approach.
And collaborative science, yeah.
Andy, thanks so much for your time. We're going to have to leave it there. You can find the rest of the podcast at our website. That's www.topmedtalk.com. And you'll also find some embedded material related to the expeditions and their work on hypoxia. For now, that's all from us, but we look forward for you listening soon. Many thanks.
Top Med Talk.